Hi, my name is Paola Vermeer, and I'm going to present to you my poster. My lab has been working on the process of tumor innervation, and we've been particularly interested in understanding what these intratumoral nerves do once they infiltrate the tumor bed. It has now been widely accepted that nerves are actively recruited to peripheral cancers. We have previously shown this by immunohistochemical staining of patient samples for neuronal markers. What we have done now is trying to understand whether these intratumoral nerves remain functional, and if so, how do they contribute to disease progression? In this poster, we have used a number of different methods to try to address this very important question. Initially, we looked at, we used bioinformatics to assess whether or not tumors have a higher incidence of expression of neuronal genes. So for example, here what we did was using a combination of bioinformatic analysis from the Cancer Genome Atlas and OncoLink databases. We simply asked whether cases of ovarian cancer that expressed neuronal genes correlated with how those patients did. Did they have a poor or good survival? And what you're looking at here in this heat map is these neuronal genes that were analyzed for ovarian cancers. And as what I hope you can appreciate is if those patient samples had a high expression of neuronal genes, they were correlated with poor overall survival. Whereas if they had a lower expression of neuronal genes, those patients had a better survival. So this suggested to us that the expression of neuronal genes may actually have a functional consequence to the patient. And when we further analyzed these data sets by looking at pathways that were driven by these genes, we found that both the molecular function and the cellular component pathways were generally associated with neuronal functions, such as calcium cation channel activity, ion transmembrane transporter activity, neuronal cell body activity, and the like, suggesting that, again, these neuronal genes weren't just upregulated and didn't have a function, but in fact, they were actually activating signaling pathways in the cancer, and this correlated with poor outcome in the patients. What we then did was we asked whether or not these intratumoral nerves had functional connections at the tumor bed. And we did this using a methodology called proximity ligation assay. And proximity ligation assay is very similar to immunofluorescence in that you use antibodies that will target your two proteins of interest. And you're asking the question, are these two proteins in close enough proximity to each other to physically interact? And so very similar to co-immunofluorescence, the first step is to bind your two proteins of interest with antibodies specific to them, so they will be recognized. And the difference comes in the secondary antibody. So unlike with co-immunofluorescence, where your secondary antibody is tagged to a fluorophore, for proximity ligation assay, your secondary antibody actually has a little piece of DNA, a little oligo on the end, so that if your two proteins of interest are in close enough proximity to each other, these two pieces of oligos will be able to be annealed. And then you can induce rolling circle amplification, which is then linked to a fluorophore. And so the end result is if your two proteins of interest are in close enough proximity, you will see a dot of fluorescence. If they're not, your two pieces of oligos of your DNA will not be annealed, you will not induce rolling circle amplification, and you will get no signal. So it's really an all or none. And so what I'm showing you here in the case of high-grade serous ovarian carcinoma, these red dots are PLA positive puncta. And when you compare and quantify the amount of PLA positivity in the high-grade serous sample versus normal ovary, you can see here that there was a significant increase in these puncta, suggesting that at the tumor bed, these tumor infiltrating nerves may actually form synapse-like structures. And so if that's true, then we reasoned that within the tumor bed, then we should be able to measure electrical activity if these intratumoral nerves remain functional. And so we did this by using a method called microelectrode array. And this has been used for many years in the context of neuroscience, where they're studying connectivity of slices of mouse brain. And so what MEAs are these custom-made wells, essentially, that have impregnated in them multiple microelectrodes that you can order in different configurations. And so what you do is you take your brain slice, or in our case, a tumor slice, and these are fresh tissues, these are not fixed, and that you overlay them over the electrodes. And so they make physical contact with each other. And what that then allows you to do is to measure the electrical activity of that slice. So whatever was captured within that slice, if there were active neurons in there, you should be able to get an electrical readout from them. And so what we did was we collected samples from patients that had high-grade serous ovarian carcinoma or benign gynecologic diseases. These were typically benign cystadenomas, and we cut the tumor slices into the tumor samples into multiple slices, and then ran them through microelectrode array analysis, and just really looked to see whether there was a difference in electrical activity that we measured from the malignant disease versus the benign disease. And so what I'm showing you here are the data that were generated from multiple different patient samples and slices from each sample, showing that both the baseline and the evoked electrical activity was significantly higher if the tumor was a high-grade serous ovarian carcinoma as opposed to a benign gynecological tumor. And what I'm not showing you here was that we found that the electrical activity from the benign slices was no different than that from normal ovary, for example, suggesting that there was something different about the malignancy. It was either in the number of nerves that were infiltrating the tumor bed, or maybe not so much the number of nerves, but the complexity of the connections that were being formed at the tumor bed were higher in the context of the malignancy, so that the electrical activity that we were able to measure was significantly higher. So this told us that these tumor infiltrating nerves remained active at the tumor bed. But really, to further define this, we decided to implant ovarian tumors into 2 different mice. Our control animals were just wild type C57 black 6 female mice. And then we used a double transgenic mouse model, which I can show you here how it was generated. We crossed a TRPV1 Cree mouse and the Rosa 26 diphtheria toxin A mouse. And so what you do when you cross these 2 mice is you generate a TRPV1 DTA mouse, which means that all TRPV1 expressing cells, including neurons, are ablated because they start to express diphtheria toxin. And so here what you're looking at are just examples of dorsal root ganglia that were isolated from the TRPV1 DTA mouse or the control mouse that were immunohistochemically stained for TRPV1. And as I hope you can appreciate, in the control DRG, you see TRPV1 positive immunostaining, whereas in the TRPV1 DTA, it is gone, confirming that the TRPV1 DTA mouse is lacking these nerves, which we had previously characterized are the subset of sensory nerves that infiltrate high-grade serous ovarian carcinomas. And so what we did was we implanted mice with these tumors and harvested the tumors, fresh tumors, and did microelectrode array analysis, and found that there was a significant difference in the electrical activity that we were able to measure from tumors that came. These are the same tumors, just implanted in C57 black 6 or TRPV1 DTA, and there was a significant reduction in electrical activity if the tumors were implanted in a TRPV1 DTA mouse. So that really confirmed to us that if you are able to, in our case, remove these tumor infiltrating nerves from the mouse, the electrical activity is dampened, suggesting that what we were measuring was at least in part coming from actively recruited nerves that were in the tumor bed. And finally, what we wanted to ask was, so we can appreciate that the tumor infiltrating nerves remain functional, right? We can measure this electrical activity. We show that we have increased PLA positivity in the malignancies. And if we remove the nerves, we can decrease the electrical activity. But is there a consequence for disease progression? So we first asked the question of whether or not, in the context of an innervated tumor that's innervated with sensory nerves, can the tumor cells respond to any of the neuropeptides, neuroligands that are being released by these active, which we've now shown are active nerves. And so one of the major neuropeptides that are released by these TRPV1 positive sensory nerves is called substance P. And substance P's main receptor is called NK1R. So we just did straight up Western blot asking whether ovarian cancer cells express the NK1R receptor. And what I'm showing you here is that of all the ovarian cancer cells and even the control fallopian tube epithelial cells that we tested, they all express the NK1R receptor. So if that's true, then given that these tumor infiltrating nerves remain active, one would imagine that at some level they're releasing substance P into the tumor environment. So what is the consequence on the tumor cells? And so we asked the simple question, and we did this in vitro, of what happens to cellular proliferation of ovarian cancer cells when they're exposed to increasing concentrations of substance P. And we looked at this just by a simple proliferation assay, both at 24 and 48 hours. And we found that the high-grade serous ovarian carcinoma cells respond to substance P with significant increases in cellular proliferation. And to determine whether this was a direct effect of substance P binding NK1R expressed on the tumor cells, we repeated the assay, but now we added increasing concentrations of an NK1R antagonist. And now we can see a dose-dependent decrease in the proliferation of the cells, confirming that this was in fact mediated by substance P binding to NK1R. And another component of disease progression is obviously cellular migration, right? When cells leave the primary tumor and then migrate and take up residence in a distant organ and metastasize. And so we did a simple transwell assay where we put the ovarian cancer cells on top of a semi-permeable transwell membrane and then treated with substance P in the basolateral well. And we see that there is an increase in cellular migration across that membrane that we can block if we also include the anti-NK1R antagonist. So this suggested that substance P released by tumor infiltrating nerves was functioning to promote disease progression, not just by inducing cellular proliferation, but also by inducing migration. And so our conclusions are that high-grade serous ovarian carcinomas are highly innervated. They harbor expression of neuronal genes, and the higher the expression of neuronal genes, the poorer the survival for cancer patients. High-grade serous ovarian carcinomas also have elevated electrical activity that we can measure. And it seems to stem, at least in part, from TRPV1 expressing tumor infiltrating nerves. Substance P can promote not just cellular proliferation, but also cellular migration, which actually suggests that either the substance P becomes a therapeutic target. So if you can block it, presumably proliferation and migration would significantly decrease, or you can block the receptor, right? Like using an NK1R antagonist. So there are therapeutic targets that we can now go after and see if we can alleviate some of these effects. And so I'd like to thank all my funding sources, especially the DaCCoTA, for having this wonderful symposium and also for the funding for all these years. Thank you very much for your attention.